So this is a case. This was a case in Indiana that I testified in Vanderburgh County. Javier Lugo was the prosecutor, and there was a 4-person DNA mixture on a clear plastic baggie that was found inside of a cracker box in a prison. The defendant was allegedly dealing in cocaine, and they found 21 grams of crack cocaine. The picture is a picture of the lab test that was done to show that there was cocaine present. There was a Daubert hearing, and at the Daubert hearing, there's the usual Daubert axes of establishing, is there an error rate, is the method testable, has there been peer review, and is it generally accepted? There are actually 10 prongs of details that are listed in some Indiana evidence book that Javi went through. The main thing is validation. Honestly, if you have validated the system and published it, you've pretty much established all 4 prongs of Daubert because it's testable. You've established an error rate if you've done that in the validation, and you've published it, and it's been accepted by the scientific community. The main validation axes that we've done for 10 years and are recommended in guidelines, for example, some recent guidelines last year from SWGDAM, are sensitivity, which is that you're not falsely including people, specificity, which is more important for a court, that you're not falsely including people, you're not finding innocent defendants guilty, and reproducibility. With complex algorithms that may have 100 dimensions that they're thinking about, randomized algorithms solve those problems, and they're not expected to give identical answers every time. They're usually within a factor of 10. You want to make sure that the answers are close. So these are the sorts of axes that are tested. There has been widespread acceptance of TrueAllele. These are some of the states and countries where TrueAllele has been admitted, from California through Virginia and overseas. There are 5 crime labs currently using the TrueAllele. Actually, there's 6 crime labs. There's 2 in South Carolina. And these were the first labs to ever use probabilistic genotyping and bring it on board and bring it into court. TrueAllele has been used in over 500 criminal cases in 35 of the 50 states. That's over 2/3 for both prosecution and defense. And TrueAllele was used 10 years ago to reanalyze the victim remains in the World Trade Center disaster, reassociating them with missing people. The judge was persuaded, and he accepted TrueAllele into evidence, finding that it's scientifically reliable as a method of DNA evidence interpretation. So we had shown other slides before this to the judge and the jury, but I'm continuing now with what we showed the jury in a more leisurely way. On the left is an evidence item that the cartoon represents a mixture. There's 2 or more people's DNA mixed together in the sample. The lab does exactly the same work. They extract the DNA, they amplify it, and they separate, and they generate a signal, which you see in the center. This is what evidence data looks like in this cartoon example, but notice there are now 3 allele peaks, not 2, and since people have only 2 parents and they get 1 allele from each, there's another person in here, because people can't have more than 2 alleles most of the time. So what's happened? Suppose that we already know that there's 1 person who has a 10-12, as we saw previously. Is there a genotype? The question is, what's the genotype of the second person that we're trying to separate out from the mixture? What's new here is that there's an 11 allele that wasn't in the data before, and we don't see anything else that's new. There's no 8 or 9 or 13 or 14. So essentially, that 11 can combine with anything else. A 10 and 11, an 11 and 11, an 11 and 12. Any of this is possible. And when you write down the list, as you see on the top right, it isn't just 1 possibility anymore. The 100 possibilities of allele pairs, different genotype values, has been condensed by the data now to just 3 possibilities. It'll generate a lot of information, but there's no one unique choice. And in science, when there's multiple choices, there are methods that will put probabilities to those different options. And that's what TrueAllele and other probabilistic genotyping software does, which is why it's called probabilistic genotyping. There's a list of possible values with probabilities. When a comparison is made later on to a known genotype, say an 11 and a 12, because TrueAllele has separated out the genotype, it's just like old DNA matching from 15 years ago, except there's a reduced strength of match. By how much? If the probability is 50% of an 11-12 in the evidence, that will reduce by 50% or have the match statistic at that location. And you'll see that visually in just a minute. Unlike the buzzsaw approach, computers can actually use all the data. This is the data pattern in this case at location of chromosome 5. And here we see that the different alleles, the events, the 10, 11, 12, 13, and 14, indicated on the boxes on the top. That's the different number of repeats. The axis on the bottom is the length of the paragraph going from left to right. As the length increases, the number of repeats increase as the size increases. The height is fluorescence. RFU means relative fluorescence units, and it's a measure of how much DNA is present at each allele. Some alleles, like at 12 in the center, are up at 1500. They have more DNA than its neighbor to the left at 11, which is down at 500. It has roughly 3 times as much DNA. So using quantitative data means that the peak heights are considered. It also means that the pattern is used. The computer is going to have to explain the pattern, and variation information can be extracted statistically as well. This is how the computer thinks. What it can do, because it has a lot of math and it knows how to model uncertainty, but also because it's a computer, so it can try out 100,000 different possibilities in a reasonable amount of time, is it is going to propose 100,000 different solutions of what the genotypes can be. We tell the computer, run for 100,000 sampling cycles, and let's assume that there's 4 people present. We can give it different assumptions. Once there are enough, the answers don't change. So suppose in blue that the first person's allele pair is a 12-13. We see that as 2 blue rectangles shown in the center that are pretty tall. And then that there's a second person's allele pair shown in orange that's an 11 and a 12. That's added on top. So the 12 location now has an orange and a blue from the first and from the second person. And then there's a third person's allele pair in green, adding some more, and a fourth person's in purple. But what you're seeing is that this particular proposal, 1 out of 100,000, adds up to heights that give a pattern, if you look at the tops of those rectangles, that look like the data. This is a good explanation in that it fits the data. It explains the data. And that confers higher probability to each of those 4 genotypes, the blue, the orange, the green, and the purple, or 1, 2, 3, and 4. The computer's also trying out many, many more possibilities that don't look like this data pattern at all. For example, if you took the 13 in blue and moved it over to 15, then you've got a big peak at 13 that's not explained, and you've got something that you're explaining at 15 that isn't there. And that would have lower probability. And so by trying out many possibilities, the computer samples the space of solutions and can assign probabilities. This is what a genotype looks like in the real world when there's uncertainty. There were 15 different STR loci that were tested, different genetic locations, and there were 4 different contributors that the computer's trying to separate out. So 15 locations times 4 contributors is 60 genotypes. This is one of those 60 genotypes. This corresponds to that blue contributor that we saw, the first one, at the location D5. And this is what a genotype is. It's a list of possibilities with probabilities attached to them based on the data. So if you look at the horizontal axis, you see allele pairs, the different values, a 10-11, 11-11, 10-12. That's a listing of about 10 of 100 possibilities. This is where more probability has been concentrated and other probability has been removed from elsewhere. The vertical axis is probability, and these numbers add up to just under 100% because there is probability elsewhere. This is what it looks like. 10-11 is a 1% probability, 11-11 is a 5% probability. If you read from left to right, this is a visual representation of a genotype. This is objectively determined solely from the data. The computer has not looked at the defendant's profile or genotype, has absolutely no idea what you want. It's just doing a separation based on the data. Afterwards, a comparison can be made to get match information. First, if it's one locus, then we'll look at others. And the question we're trying to ask is how much more does the suspect or the defendant match the evidence than a random person? And it's a ratio. All match statistics in DNA or forensics are ratios. And the ratios can be expressed in about 10 different ways with different words, different formulas. They're always ratio of probabilities, and they all give the same number if you're using the same method. They're just different ways of writing down the math. Here we're looking at the probabilities of genotypes. As we saw before, the evidence gave a 43% probability at 1213, where the red circle is, and the blue bars are the same, that's the evidence. The brown bars is the chance of a coincidence. What's the chance that somebody in this room, by chance, would have one of these genotypes? For example, the chance that you would have a 1213, or somebody taken at random, would be about 17%, about 1 in 6. What a match statistic is a ratio of those probabilities, a ratio of the evidence genotype, what we saw after looking at the data, that's 43% in blue, divided by what we thought before, which is 17% in brown for a random person. So what's the chance of a match divided by the chance of a coincidence? And for a person with a 1213 genotype, that number is a little over 2, 43 divided by 17. If you were to move one to the left and look at the 1113, the evidence in blue is half the height of coincidence in brown. That would give an exclusionary value of a half. The computer doesn't care which one you pick, but it's already calculated everything you need to make a comparison and determine inclusionary or exclusionary weight. Listed here vertically are all 15 loci, and horizontally on a bar chart is the match statistic, also called likelihood ratio, and that's on a scale of 0 to 200. The value of 2 that we just looked at on the 5th chromosome is circled in red. That's around 2. Some numbers are much larger, like out at 50 or over 150. Others are smaller, and the length the bars are showing you that. Multiplying these numbers together gives you the total match statistic because they're independent locations, and that independence lets you multiply them together. And this is what you end up with. And I think what's most important here is the language. This language is as simple as DNA has ever been for the last 20 years because it talks about one separated contributor of the 4 from the bag relative to one person, that's the comparison, and that's with respect to a random genotype. So it's what we've always done, except instead of having 100% probability from evidence, now there's only 43% probability. But we're comparing one genotype with one genotype relative to random, and the language is English that comes right off the math. Words like match, probable, multiplying, coincidence, this is the actual math. And it's fairly straightforward. It says a match between the bag and Mr. Forrest is about 25 trillion times more probable than a coincidence. And that's language that lawyers and juries can actually understand. The computer's done a lot of work to bring it to that point. The match statistics as we presented at trial are shown in a table. The rows are from the bag with the white substance, believed to be crack cocaine. There was another piece of evidence that was a foil package. That's in the second row. And then there were 3 people that was compared to plus a lab analyst. And TrueAllele doesn't really care if there's contamination. It just says, okay, so now there's 4 contributors instead of 3. And as you can see, Mr. Forrest is matched against with a stat of around 25 trillion. The analyst is accounted for with a stat of in the thousands. And on the foil package from the cracker box, the stat is 100 quintillion. To help with what's going to happen next, I'm going to represent these numbers as the number of zeros, because that's how information is looked at. And we'll just look at one of them. If you look at the number 25 trillion, 25 trillion is 25 followed by 12 zeros, or 2.5 followed by 13 zeros. So in other words, that's a 1 followed by 13 zeros. And if you look in the top left corner, you'll see that number 13.4. It's called the logarithm, It's basically the number of zeros in the match statistic. It's easier to work with numbers like 13 or 26 than it is to remember what all these words are and count. We're just counting up the number of zeros. And Mr. Forrest was convicted at trial, and there were some aggravating factors for his possession. He was sentenced to 11 years in prison.